Good morning. Good Wednesday morning. Good morning, everyone. Good to see you.
Morning.
Morning.
All right. If you would, please go ahead and type your first name or your nickname into chat for me. That'll be my record of your attendance today. Thank you. Good to see everybody. Very good. Just about everybody's here. Still waiting for John. Poor John, he's the only male among a group of tremendous females.
He's used to it. It's like that in the nursing program.
That's good.
He hangs in well.
That's good. Well, that's very important. I told him on the discussion board, I told him how wonderful it was that he was pursuing nursing, because we need more men in nursing. And it's wonderful to see. And I'm sure you ladies will keep him in line. All right, very good.
He was in my clinicals, and he's going to make a very good nurse.
Oh, that's wonderful. That's great to hear. All right. So
he does often have like technical difficulties. I will tell you that his reception, like his Wi-Fi at his house, kind of sucks.
Oh, that's too bad. Yeah, I understand that. You know, we all have we all live in different locations, and the Wi-Fi access just varies tremendously. As you know, the Wi-Fi access is just it really needs addressing here in the state. We need to get on top of making sure that everybody throughout the state has access to really strong Wi-Fi. All right. So today is our second meeting, our first official lab day. And today we're going to be talking about two subjects. We're going to be discussing biosafety in the microbiology lab, and in fact, in all labs that handle potentially dangerous materials. And we're also going to be talking about microscopes. We're going to be talking about the care and the use of a compound light microscope. So first things first, let me ask if anybody has any questions about anything. Feel free to type them into chat. I know we covered a lot of material when we were together on Monday. We talked a lot of housekeeping things about the course and about the technology we're using, Canvas and Zoom and YouTube. And if anybody has any questions or problems that have come up, we can certainly talk about them at the beginning of our lab times together. You also have started into your first lecture topics, which are Introduction to the Microbes and the history of microbiology and microscopy. So this is also a good time to bring questions forward. If you have any questions about any of the material in lecture videos or in anything you're reading, feel free to do that. I can see on Canvas that several of you have already completed the first lecture topic and have taken the quiz. Remember, that quiz is due tonight by midnight. So if you haven't already taken the first lecture quiz, Intro to the Microbes, just make sure you get that completed by midnight tonight. I was very pleased to see that those of you who have taken it did very well on it. So that's a great start. And again, the quizzes will give you an opportunity to see the way I write questions. Primarily multiple choice questions, but you can see I also do a thing with, I make true false questions into multiple choice questions sometimes. And again, sometimes there will be short answer questions. So I was glad, like I said, I was glad to see that people who have taken the quiz so far have done well on it. So no questions so far. If any come up while we're talking, feel free to type them into chat. I do check the chat periodically. So don't hesitate to do that. This is our time together, and this is the time for us to talk about things. And I really do believe that if you have a question about something, one of your classmates probably has the exact same question. So don't ever hesitate. We're all colleagues here. We're all professional colleagues here, and there's no need to be nervous or embarrassed about asking questions. All right, so looking ahead, the other thing I do in laboratory, particularly on Wednesdays, is I just try to remind us all what's coming up, what we're doing this week, and what's coming up next week. So I'm going to go ahead and pull up our Canvas course page. You bear with me just a minute. That's what you should be seeing on your screen right now. You should see the home page for our Canvas course, Summer 2021. So I'm just going to go over here into the navigation menu on the left. I'm going to click where it says modules, and I'm just going to scroll down to this week. So again, the first quiz that you're taking is due by tonight at midnight. And then the next quiz you have will be due by Friday at midnight. And here's today's lab. Here are the slides that we're going to be using today to talk about biosafety. We're also going to be watching a video together that I made for us about how we care for and use the microscope. I've posted this tutorial sheet for you regarding the microscope, the typical compound light microscope. We all come to class with different experiences, and I know some of us have more experience handling microscopes than other people do. So we're going to talk about the scope today, and this is a nice little tutorial sheet that you can use to supplement what we talk about in lab today regarding the microscope. And then finally, you do have lab homework this week, your first lab homework assignment. This is due by Sunday at midnight, and it will cover the material that we talk about today. Looking ahead to next week, here's your to-do list. I know it's hard to imagine, but you have your first exam next week. You have an exam a week from today. And that exam will cover the first 3 lecture topics. So you have lecture exams in here, and you have lab exams in here, or what we call lab practical exams. So this lecture exam is going to cover 3 subjects. It's going to cover the Introduction to the Microbes lecture. It's going to cover history and microscopy. And it's going to cover the cell, which is the lecture topic for next week. Now, one thing I want to point out, this exam takes place on Wednesday next week. So that means you've got to get this lecture under your belt before Wednesday. You've got to watch the lecture videos and you've got to take the lecture quiz before Wednesday. The quiz is actually set to be due by Tuesday night at midnight. That's unusual. Usually you have lecture quizzes due Wednesday and Friday. But next week, because of the exam, your lecture quiz is due by Tuesday at midnight. Lecture exam takes place on Wednesday. Again, I'm not going to spend time talking about the exam today, but we'll talk about it more on Monday when we get together. And when we get together next week on Monday, we're going to be talking about examination of living microbes. We will also have lab on Wednesday, and we're going to be talking about what's called simple staining. So that's the week we have ahead. Remember, you always have this to-do list. And this list gives you all of the information you need about what's happening in that week. So do make use of that. Make use of that to-do list each week to help you stay on track. All right. Welcome, John. We're glad to see you. Oh my gosh, computer updates. Yes. The last time my computer did an auto update on me, and I use a MacBook, and the last time my computer did an auto update, I think it took an hour. And I was so angry because I didn't even have a chance to decide whether or not I wanted it to update. And it just did it, and I couldn't use my computer for a solid hour. So
yeah, that's exactly how it went.
Yeah. Elaine is asking about the exam. Once we open the exam, how much time do we have to complete it? The exams, you're given 90 minutes, Elaine. And we will talk about this more on Monday, but for now, know that in this course, in my experience, most students complete the exam between about 30 and 60 minutes. So you will have more time than you need. I know many of us have a lot of anxiety around exams, and I understand that, which is why we try to prepare ourselves with quizzes and things in this course. But one thing that I strive for is to make sure that everybody has plenty of time to finish an exam. So I don't try to push you that way. So if that takes away anybody's anxiety, I'll be glad. All right. So any questions about our schedule? Any questions about what you need to get done this week? Any questions at all so far? All right. And feel free, like I said, to type into the chat as we have our lab this morning. I do check it periodically as I'm talking. So let's go ahead and jump in. I'm going to pull up our slide set for today about biosafety in the microbiology lab. And that's what you should be seeing on your screen right now, the title slide for the lab. Biosafety is actually something that's near and dear to my heart. I've been in a lot of different laboratories in my life, both in my veterinary practice and also in the research laboratories that I've worked in and teaching laboratories that I've worked in. I didn't mention it in my bio on the introductions board, but I spent 3 years as the acting chemical hygiene officer at Keene State College. And if you're not familiar with that terminology, a chemical hygiene officer works with environmental health and safety at colleges and universities and various commercial businesses, any place that handles dangerous materials or hazardous substances. So I have a real soft spot for biosafety. It's something that I care a lot about. And I think it's really important that we talk about it anytime we're taking a course like this one, where we're talking about potentially hazardous things. So that's why we include this material in this course. So some of the key points for what we'll be talking about today with regards to laboratory safety, particularly micro lab safety. Number one is door signs. If you've ever walked into a laboratory space, you may or may not have noticed this diamond on the door. All laboratories have to display what's called an NFPA Diamond. This is a system that was put together by the National Fire Protection Association, the NFPA. And this diamond does two things on the door of a laboratory. It alerts anyone who works in that laboratory what kind of hazards are in that laboratory, what kind of hazards that they are working with each day. And number two, it alerts first responders when there are issues in laboratories, when buildings catch fire, when there are explosions, things like that. Any first responder that arrived at that laboratory would be able to read the diamond and know what kind of hazards are inside. So this is something we're going to talk about. I don't expect you to memorize anything that's on this particular slide, other than the fact that these are hazard symbols. These are there to warn us about hazards. And we'll come back to the diamond in just a minute. The other key points we're going to touch on today are this one right here in capital letters: no food or drink. We don't eat and we don't drink in laboratories. You know, one of my pet peeves when I watch movies, I love movies and I watch a lot of them, is that there's sort of this running joke in Hollywood about scientists eating in laboratories. And they usually try to make it out that usually the scientist is doing something disgusting with one hand and eating a sandwich with the other. Like,
why is it always a sandwich? They're always eating a sandwich.
It's
in every
always a
movie.
sandwich. And it's so frustrating to me because it's, first of all, it's not true. And it just makes us look like we're some kind of animals or something. I don't know. Recently I watched just a rerun of an old episode of Big Bang Theory with my son. And there's a scene where this neuroscientist is standing dissecting a human brain, and she's got a sandwich in her hand. And I know they're just being funny and silly, but obviously we don't eat and drink in laboratory spaces. It's not safe to eat and drink. And if you were to do that, I mean, you would be in trouble with your supervisor. If you were to sit down in the laboratory at your workspace and pull out a sandwich, you'd be in trouble. It's not safe. We work with hazardous chemicals in laboratories. We work with materials that you could easily contaminate with food in a laboratory. And if you're working in a microbiology laboratory, you're taking a terrible risk eating and drinking. So generally speaking, what people do in the real world is they simply put their food and drink in a separate location. And sometimes a laboratory will, devote a corner of a lab, or maybe an adjacent laboratory or an adjacent closet or some other space. They'll dedicate to a place where people can step away from their work and have a snack or grab a drink. But no, you don't sit at your workbench and do your work and eat and drink with your other hand. And the reason, of course, is that when you eat and drink, you're pulling food and drink into your body, and you're potentially pulling hazardous materials down into your body. So similar to the no food and drink rule is that we don't do things that might introduce things into our body another way. So for example, it's really discouraged to do things like apply makeup or especially lipstick or lip balm in the laboratory. Again, if you pull out mascara or something and you are applying makeup near your eyes are a portal into your body. Your mouth obviously is a portal into your body. So if you pulled out lip balm and applied it to your lips, you're risking the fact that you may have chemicals or microbes or other hazardous materials on your hands, and you've just potentially introduced them into your mouth. Generally speaking, we try not to handle our face when we're working in a micro lab. We try not to handle our eyes and our nose and our mouth because those are portals of entry into your body. If you are working with microbes, you have to assume that you've got contamination on your hands. So for you to rub your eye or, put your finger in your nose or whatever, you are, again, potentially contaminating yourself. So we try really hard to learn not to touch our face while we're working. And along those same lines, we are gloved in the microbiology laboratory. We're gloved in most laboratories. Gloves are there for two reasons. They're there to prevent you from contaminating whatever it is you're working with, your skin on your hands. And they're also there to protect, I'm sorry, I said that backwards. They're there to protect your work from being contaminated by you. And they're there to protect you from being contaminated with your work. So, obviously, if you're in a hospital, some kind of diagnostic laboratory, and you're handling patient samples, you're handling specimen cups of urine, or you're handling tubes full of blood, we generally have gloved hands because those are potential sources of contamination for us. And when we open those containers and we start doing diagnostic tests on those body fluids, we also want to protect ourselves from them. So we use a lot of gloves in the micro lab. It's just a part of life for us. The other thing we're going to be talking about is waste disposal. And this goes not only to our own safety, but also to the safety of our fellow lab mates, our fellow colleagues, our community. Again, I take all of this very, very seriously. We have to manage our waste so that we are not a source of contamination in the environment, particularly. So we have to be very careful what we put down the drain, what we put into a trash can. We have to make use of waste bottles, particularly labeled for what they have inside, so that professional waste management companies can come and remove those materials from the lab and take them somewhere where they can be properly disposed of. And of course, in a microbiology lab, we also make heavy use of autoclaves. We're going to be talking about the autoclave in a couple of weeks in some detail. But for now, if you're not particularly familiar with what an autoclave is, it's a machine that we use in laboratories and in hospital spaces in order to sterilize things. So sometimes we are sterilizing materials for disposal. In other words, if I have a container that's full of living microbes, I need to sterilize that container before I throw it away. I need to kill all of those microbes before I throw it away. Other times I'm taking a reusable item and I am sterilizing it so it can be used again. Think about all those single-use items in hospitals and rehab facilities and nursing homes. Think about single-use syringes and needles, single-use catheters, IV catheters, and so on. Those things were autoclaved at some point in their life. In order to make them sterile so that they could be used. Another thing you'll hear me talk about today is the importance of hand washing. I feel like we've all had a reminder lesson about hand washing during the pandemic. but it's very important when you're working in a microbiology lab to keep in mind that your hands are a primary source of contamination, both to you and to others. So it's critical that we wash our hands. We wash our hands before we start our work, just in case we've carried anything into the lab, and we wash our hands before we leave so that we're not carrying anything out. All right, let's take a minute and look at this NFPA Diamond in a little more detail. Again, please don't write down all of these categories here, or I should say, don't write down all of these numbered descriptions here. The only thing I want you to be familiar with for this course is what the 4 categories are. You should know these categories because you're at a point in your education and in your career where you're likely going in and out of rooms that have these signs on the door, and you should know how to read them. So the top of the triangle with the red color is probably the most obvious one. This is the fire hazard square or diamond. This is the diamond that's telling us what kind of hazards are in that room regarding fire. The yellow triangle is telling us about things that are unstable in the room. Now, what do I mean about that? There are a lot of chemicals that have instability associated with them in terms of things like catching on fire or sometimes blowing up. Things that get exposed to the air sometimes will ignite, or things that get exposed to water will sometimes ignite. So that's what this diamond is for. It's letting us know if there's any sorts of chemicals in the room that have that instability about them. The white box tells us about what are called specific hazards. So these are very sort of special hazards that don't fall into other categories. If there are chemicals in use that are corrosive, for example, chemicals that are radioactive, strong acids, strong bases, things that are oxidizers, things that are likely to help other things burn. That's what an oxidizer is. That goes into the white square. And then the blue square is the health hazard square. So this is a square that tells us how hazardous the lab is, generally speaking. What kind of chemicals are in the lab that are dangerous to our health? And of course, that can range from things that are neurotoxins to things that are reproductive toxins, things that are acutely toxic. In other words, they could harm you right now, today, and things that are chronically toxic. So you can see why it's important when we work in laboratory spaces or we go in and out of laboratory spaces in our day at work that we know, am I being exposed to chemicals here in this room that over time are going to be toxic to me, could potentially impact my fertility, for example. Now, we don't just talk about the specific hazards, the 4 specific hazards, but we also give it a number. And that's the really critical part of the NFPA Diamond. In other words, with regards to fire hazard, there's going to be a number in this box hanging on the door of the laboratory. If the number is 0, or if there's just a dashed line across the box, that means there's nothing in here that is considered a fire hazard. The numbers then go up from there. The highest number you're going to see in the box is a 4, and the 4 is the highest hazard. So how do they rate fire hazard? Well, they look for what's called flash point. They look for the presence of chemicals that are going to ignite at certain temperatures. And the highest hazard for flash point is considered a chemical that is capable of igniting below 73 degrees Fahrenheit, which is essentially room temperature. So you can imagine that's a high hazard, and that would be the number 4 in the box. So as the numbers get higher, that's more hazardous. As the numbers get lower, that's less hazardous. Instability, again, depending on whether or not that item is, unstable only under certain circumstances, the number is going to be low. So chemicals, for example, that become unstable only when they're heated up, that's going to get a lower number than something that's capable of detonating spontaneously. Same over here for specific hazards. It's going to depend upon the particular danger that's associated with a specific hazard and the degree of the condition. For example, if something is corrosive, if something is acidic, how acidic is it? Are we looking at acids that have a pH of 1, that are considered strong acids, that are more hazardous? That's going to get a higher number here. Health hazards, same thing. The lower the number, the less hazardous it is. The higher the number, the more hazardous it is. And you can see that the highest number for health hazard, 4, is deadly. So there are materials in that room that if we get exposed to them on an acute basis, they can kill us. So NFPA diamonds are present on the door or adjacent to the door. So the next time you're in your workplace or you're in buildings, science buildings at school, take a look. Take a look at the diamonds as you walk around and see what numbers are present in the diamonds. They are there for us. They're there for us so that we are aware of the hazards that are in our working environment. Whether or not you actually work in that room, it's important to know. It's important to know what kind of things are around you in your workplace. All right. So we have sort of a ritual in the microbiology laboratory that we try to do every time we walk in. So whether it's a research laboratory at a college or perhaps a commercial R&D type of business, a research and development business, or perhaps a diagnostic laboratory in a hospital or in some other healthcare facility, people who work with microbes are going to follow sort of a ritual of activity each day. When you get to the laboratory, we always put our personal items away. They need to be away from your workspace, whether you're working on a bench or you're working, on a shared table space. Your personal items, your coat, your purse, your lunch, your cell phone, those kind of things get tucked away. And as I mentioned, most working laboratories have a space devoted to this. Some place that workers are allowed to deposit their personal materials. It's really important that your workspace is clean and clear. So by clear, I mean not cluttered. And by clean, I mean disinfected. We're working with microbes. So we recognize that having microbes on our workbench that we didn't put there, that we didn't purposefully put there, that could interfere with the results of our work. The 2 most common disinfectant chemicals that are used, not just in micro labs, but really in labs related to healthcare in general, are alcohol and bleach. So depending on the laboratory, and it will vary, you'll generally find them that they're using either alcohol or bleach as the disinfectant. Now, when it comes to alcohol, we use a diluted solution. We're going to talk about this in a few weeks when we talk about chemical disinfectants, but alcohol actually works better when it's diluted. I know that seems counterintuitive, but it's best to use a solution between 60 and 90%. So we dilute it to 60 to 90%. Again, during the pandemic, a lot of us have been forced to learn a little bit about disinfection. If you remember in the early days of the pandemic, people had all kinds of questions about how do I disinfect my doorknobs? How do I disinfect my grocery bags? Do I need to disinfect my boxes when they arrive from FedEx? Everybody had a lot of questions. And it's intuitive to think that if I'm using alcohol, I should use it at full strength because it's stronger, so it ought to kill better. But when it comes to alcohol, that's actually not true. You need a diluted solution to get it to work at its best. So 60 to 90% for alcohol. It doesn't matter what kind of alcohol you're using. So a lot of times we use isopropanol in the laboratory or isopropyl alcohol, but you can certainly use other alcohols as well. You can even use ethanol. You can even use the kind of alcohol that we drink to disinfect. But again, you would want it at a 60 to 90% solution. We've all seen those, the old movies, like the old Westerns, especially, where someone would get a laceration or a bad cut and would pour, a bottle of whiskey over it. Listen, it's better than nothing. It's better than nothing. So when it comes to disinfection, alcohol is a very commonly used one in laboratories. Now, the other one is bleach. And again, bleach is best used diluted. And look at the dilution. We take it all the way down to 10%. 10% chlorine bleach is the most effective dilution for disinfection. Now, in most laboratories that I've worked in my personal experience, alcohol is more commonly used than bleach is, but bleach is a perfectly good disinfectant. And one of the reasons that people choose bleach over alcohol is because bleach is a lot less expensive, believe it or not. So if you're in a healthcare setting and you're using a lot of disinfectant, if you're in a microbiology lab and you're using a lot of disinfectant, sometimes bleach is the more sensible disinfectant with regards to cost. Always gather all your supplies before you start working. So clear off your workspace, disinfect your workspace, and then get everything needed that you're going to be using for whatever task is at hand. The last thing you want to happen is for you to get started on a task in the laboratory, and then halfway through, you realize you don't have something you need. You need to get it all first, and you need to know what's in front of you. So that's why the next thing is so important. And I have this in capital letters because it is so important. We need to label all of our containers. This is something that's also very important in a microbiology lab. Any container that you are making, that you are producing yourself, whether it's a Petri dish or a test tube or any other kind of container, you have got to label it. We generally put the following things on container labels: our name, or at least our initials, the date that the container was made, the name of the microbe that is in or on the container, and the name of the medium, the name of the material that's in the container that the microbe is in. Sometimes those media are solid things like agar. If you're not familiar with agar, we're going to talk about it a lot this semester. It's a naturally occurring material that we use in microbiology for growing microbes. It actually comes from algae, and it produces almost a jello, a gelatinous sort of material that we can spread microbes on. So if I'm using a particular type of media in my agar, I need to label that. I need to say that this is a particular variety of media. We also use a lot of what we call broth media, which is simply liquid media that we grow our microbes in. A lot of broth media looks exactly the same. In other words, they're very different varieties of broth, but they all have a beige color. So there's no way to look at the tube and know which broth is in there. So it's up to us. We have to label it. Now, the label is there not only so that we can keep our containers separate from each other and we know what's in our containers, but it's also for our colleagues. So that if someone goes to, let's say, the incubator, where we grow microbes in the laboratory, or the refrigerator, where we're storing microbes, anybody can pick up any container and they can know what's in it. All right, it's really, really important. Listen, it's important to label containers in all laboratories. It's important to label containers in all laboratories. And you know that, you've been taught that with regards to patient samples. You have to have labels on everything. But in a micro lab, it's especially important because we're working with living organisms. And we need to be able to know what is growing in a container in the laboratory. All right. I also want to say a couple of words about what we call lab hygiene. This is important, again, really in all laboratory spaces, but it's especially important in a micro lab. You have to have good lab hygiene. Now, what does that mean? First thing I've already mentioned is keeping your workplace free of your personal items. So you should not have your purse on your workbench. You should not have your bag, your lunch bag on your workplace. You shouldn't have a cup of coffee on your workbench. It should be clear of all your personal items. You need to have your workspace defined for work. It needs to be a clean and clear place for you to work in. You need to wear appropriate clothing in the laboratory. Now that involves not only the literal clothing on your body, but also jewelry. And again, this really applies to any laboratory space. But in general, depending on where you're working, your supervisor is going to tell you what clothing is appropriate and what clothing is not appropriate. In this picture, you can see this person is wearing a lab coat. And certainly in a lot of healthcare spaces, this is what people wear. They wear their street clothes to work and then they simply cover up with a lab coat. In plenty of laboratories, people are wearing scrubs, particularly in a healthcare setting. The idea is that you've got some kind of covering over your street clothes that is going to prevent you from getting your street clothes contaminated or even damaged with the chemicals that you're using in the lab. Now, not all laboratory spaces require that you wear a lab coat. Plenty of people wear their street clothes working in laboratories. So it's especially important that they choose appropriate street clothes. Now, I'm not talking about, are they showing too much skin or something? I'm talking about street clothes that are appropriate for the hazards of the laboratory. You don't want to wear loose, flowing clothing in a laboratory, especially important for women. You don't want to wear sleeves that are wide and that are flowy and draping. I'm thinking about those bell sleeves that are sometimes, fashionable on blouses and things. You want your clothing to be fairly close to your body. You're going to be moving your arms across your work surface. You're going to be moving your body close to your work surface. You don't want your clothing to accidentally brush across your work surface. So you have to think about what you choose to wear in the lab. Regarding jewelry, same thing. You don't want to wear long, clunky, heavy jewelry in the laboratory. You don't want a long necklace, for example, that when you lean forward across your workspace, your necklace is going to drag across your bench. You don't want a long, clunky, loose bracelet on. You don't want any sort of jewelry that could potentially accidentally come in contact with your workspace. We always need to wear appropriate shoes. Again, this goes for all laboratories. Just as a general rule, we keep our feet covered. We don't wear sandals It's just not a good practice. It's not a good idea to have large portions of skin open on your feet. People drop things all the time, and it's important that we keep our feet covered to protect them. If you're like me and you have long hair, you've got to keep your hair managed in the laboratory. That generally means just pulling it back either with some kind of clip or clips, perhaps a ponytail holder. When you're at work in the laboratory, you've got to manage your hair. Your hair is a source of contamination. Your hair, number one, tends to fall out. Hairs fall off of our heads every day. Having your hair pulled back is going to help keep those loose hairs off of your workspace. The other important thing about hair is the fact that it is flammable. And especially if you're working in a laboratory that uses open flame, and open flame is common in microbiology labs, you have got to keep your hair away from your workspace. I have never experienced this personally, but I have heard dozens of stories about people who accidentally set their hair on fire. You can imagine it is terrifying. It can then, of course, lead to you panicking and your clothing gets set on fire. Keep your hair back when you're in the laboratory. So John has an interesting question here. He says, sometimes bleach is the only thing that can kill certain things like C. diff. That's a really interesting point, John. Believe it or not, every disinfectant has a list of what it kills at what concentration. So bleach, alcohol, and all the other disinfectants. And again, we're going to talk about those in detail in a few weeks. There are tables and tables, charts in textbooks and online that give us really critical information about how the concentration we need to use and the amount of contact time we need to use to kill certain pathogens. So as a very general statement, we say, yeah, we need alcohol at 60 to 90%. But, you know, it's a big difference if you're just trying to disinfect your workbench versus if you're trying to kill a pathogen that you handle routinely in a laboratory, something like Clostridium difficile or C. diff. So yeah, it really depends on your laboratory, and different labs choose different disinfectants, sometimes for financial reasons, sometimes for ease of use, and sometimes because they're handling or they expect to be dealing with a particular pathogen that's a little harder to kill. I always tell people, you know, when you go to the grocery store and you pick up a disinfectant, you know, there are hundreds of them for sale in the grocery store aisles. What most people do, right? Most people take the bottle home. They don't read the back, right? They, let's say they're trying to disinfect a kitchen counter. They're going to spray it on the kitchen counter and they're going to wipe it off, right? That's what people do. When in reality, you're probably supposed to spray it on and then leave it for some period of time and then wipe it off. But if you don't read the back of the bottle, you wouldn't know that. So you have to know your disinfectant. You're going to hear me say that a lot. You have to know your chemical, and you have to know how to use it based upon the kinds of microbes that you might be trying to kill in your lab. And if you're a lab that is handling patient specimens that likely have things like C. diff or MRSA or things like that, you need to know how to disinfect properly, because the last thing you would want to do is contaminate your workers or anyone else with such dangerous pathogens. Marilyn's saying that I'll never forget a news story where a manager was using various types of disinfectants to clean. He mixed them all together and he died. Oh, girl, wasn't that like last year? Didn't we hear a story about that last year? Where it was in a restaurant, they were mixing disinfectants. I think one worker fell unconscious. The other workers left the restaurant, smart. And then the manager went back in because the manager was trying to help and the manager died. Oh my gosh, chemicals are dangerous. And yeah, the other story that we hear a lot about, you'll hear like once a year, you'll hear a story in the news about somebody who mixed ammonia and bleach, right? Those two chemicals, when you mix them together, produce a toxic gas that will kill you in minutes. And like I said, every year there's at least one story of some person who thought, well, if ammonia works well and bleach works well, surely the two of them together will work better. And they mix them together and they create toxic amine gas and end up killing themselves. So yeah, chemicals are very dangerous, some of them. And in the workplace, workers are not always aware of hazards, which is why we work so hard in environmental health and safety circles to make sure they have access to information. But unfortunately, yeah, those stories come in the news all the time. It's very sad, very, very sad. All right. The other thing we've all had an opportunity to learn a lot more about, maybe more than we ever wanted to know during this pandemic, is personal protective equipment. Of course, there was a terrible shortage of PPE early in the pandemic, not only here in the United States, but around the world. And of course, there has been endless discussion in the public, at least, about masks and whether or not masks work and blah, blah, blah. So obviously, we use a great deal of PPE in laboratory spaces because it does keep us safe, not only from potential chemical hazards, but of course from microbial hazards as well. When it comes to the microbiology laboratory, we certainly have multiple kinds of PPE available to us. It's going to depend upon the microbes that you're handling, what kind of PPE people will be wearing. So again, things like lab coats, goggles or eye protection, various types of face masks, they're used when they're needed. So in most microbiology laboratories, that are not handling particularly dangerous pathogens, the PPE is kept to a minimum. In fact, the only piece of PPE that is always used is gloves. Gloves are worn regardless of the biosafety level that you're working with in a laboratory. You can see this term right here, BSL, that stands for biosafety level. All laboratory spaces that are working with microbes or even living cells, just living organisms, are going to be assigned a BSL level or a biosafety level. We'll talk about that more in just a couple minutes. So gloves, always, always. And as you all already know, I'm sure, gloves have to be put on and taken off correctly when you're handling potentially contaminated materials, contaminated either with body fluids or contaminated with known microbial cultures. You have to put them on correctly, you have to take them off correctly to protect yourself. Now, we don't use sterile gloves in the laboratory. That would be a waste of money. So we just buy regular old non-sterile boxes of nitrile gloves, sometimes other materials. There's a real movement away from latex gloves because of the problem of latex allergy. Couple of key points that you may or may not already know, depending upon your where you are in your educational program and where you are in your work experience. When you are wearing gloves, especially in a microbiology laboratory, we do not touch anything while we are gloved that is not routinely disinfected. Now, what do I mean by that? When you have a pair of gloves on, you assume that they are contaminated when you work in a microbiology lab. Even if you just put them on, let's say you just got to work, you just put your personal items away, you just cleared off your workspace and you disinfected it, and then you put on your first pair of gloves. Once they're on your hands, you assume that they are covered in microbes and you act accordingly. Now, it's perfectly okay for you to touch the things that you're going to be working with, but while you are gloved, you don't touch things like doorknobs. You don't touch telephones. You don't touch your hair. You don't touch your cell phone or your computer while you are gloved. You don't touch microscopes while you are gloved. Those are things that are not routinely disinfected. Listen, your workspace is going to be disinfected every day, maybe multiple times a day. And all those materials that you're handling in the laboratory, if they're not going to be disinfected, they're going to be discarded. But things like equipment, large pieces of equipment that do not get disinfected routinely, you just don't touch them when you're gloved. So what do you do if you need to touch your computer? What if you're doing some diagnostic testing and you need to enter some results into the computer? You just take your gloves off. You take your gloves off. Step away from the workbench, remove your gloves, work on the computer. Step away from the workbench, remove your gloves, work on the microscope. Remember, as soon as you take a pair of gloves off in the microbiology laboratory, you throw them away. They are single-use items. We never take a pair of gloves off and then put them back on. Remember, we're going to assume that they are contaminated. We also do not leave the laboratory while we are gloved. So if you need to go to the restroom or you need to go out of the room to have your lunch or a cup of coffee, gloves off. Remember, you're not going to touch that doorknob with your gloves on because your gloves are contaminated. At least that's what we're going to assume. We're going to always assume our hands, our gloves are contaminated. One of the things that's hardest for students to get accustomed to when they work in a microbiology lab is how many gloves we go through. I've had students put gloves on and then start to do their work, and then they need to get something out of their backpack. And I will see them sort of gingerly trying to take the gloves off so that they can use them again, because they know they haven't touched anything contaminated. And I always bark at them in the lab and I say, don't you put those gloves back on. You throw them in the trash and get a new pair. You have to get in the habit. You have to get in habits in the micro lab that may feel strange to you. But again, it's all about biosafety. You always just take them off. If you have any question about whether or not you can touch something while you're gloved, you just take them off, throw them away, do what you need to do, and then get a fresh pair. We go through a lot of gloves in the micro lab. We go through a lot of gloves. But again, it's all in the name of safety. Oh, Niani's saying that in the lab where she has worked, while they're working during the day, they're even disinfecting their gloves. Listen, if you're working in a space where you're handling, you know that you're handling significant pathogens, So if, particularly in diagnostic laboratories where you're handling urine and you're handling maybe fecal material and you're handling blood, depending on the type of testing that's going on in the lab, it's not a bad practice to, even with gloved hands, to routinely disinfect your gloved hands. Yeah, very interesting. Now, the other thing related to biosafety that I want to mention, at least briefly, is this. Every single laboratory space that you're going to go in and out of in your professional career, even if you're not working in the lab, but you are perhaps delivering samples to the lab, or you are, picking up results from a laboratory and you have to go into the room. Every laboratory is equipped with safety equipment. It's the law. And again, it depends upon the hazards in the lab. So depending on the numbers that are in that triangle on the door, you're going to find things like eye wash stations in the lab. You're going to find emergency showers in labs in case someone accidentally catches on fire. You're, of course, going to find a first aid kit. There's going to be fire extinguishers. There's going to be things like fire blankets, which are used to smother fire. It's really a good idea for you to be aware of where these things are in the laboratory. Now, why is that? Why do I need to know exactly where the emergency shower is? If you ever find yourself in a situation where there is an emergency in a laboratory, and they happen all the time, you are going to be frightened. It would be abnormal if you were not frightened. And you're going to enter into a stress response. And that will interfere with your ability to do things that sound really simple, like finding the emergency shower. So it's always a good idea to familiarize yourself just with where things are. Most of these things are labeled for people who are in an emergency situation. So for example, on the emergency shower, and we have a lot of emergency showers, for example, in Keene State in the Science Center laboratories. There's going to be big signage that literally tells you, pull this, push this, because we know you're stressed. We know you're stressed and you're in an emergency situation. But it happens that people get into these situations and they have no idea where the equipment is. I'll share with you a terrible story from UCLA that happened a few years ago. Research residents, these were doctors who were close to being done with their residency. And a woman who was also doing some research, a research project, was working in a laboratory. She was working with a material that, if it got exposed to air, would spontaneously ignite. So she was in a hood. She was in a chemical hood. There was an inert gas that was being used to keep that material safe. She accidentally removed her hand a little bit too far back into the room air, and the material caught fire. And it caught her hair on fire, and it caught her clothing on fire. All right. Now I want you to think for a minute about this person. This was a resident. This was not a medical student. This was a resident, a grown woman. She had been in healthcare for a long time. She had worked in that laboratory for a long time. She had coworkers in the room with her who were also very experienced, very knowledgeable people. And everyone panicked. And the story is, anyway, the final report that came out about the incident, people went to the sink and they started filling up cups. I'm trying to get my hand in front of the camera. Little cups of water that were at the edge of the sink. And they were throwing water on her. All right. There was an emergency shower in the room. And nobody knew. And that poor woman was burned so badly that she died within a couple of days. All right, now I don't tell you that story to gross you out. I tell you that story because that happened. That happened in a busy, high-tech research laboratory, okay? Nobody knew that there was an emergency shower on the wall. And nobody knew that she needed to be pushed into that shower and the flames put out. And she died as a consequence of that. So know where the emergency equipment is in any laboratory that you are routinely going in and out of. Listen, you may not be the one who has the emergency, but someone else in there might have the emergency and you may need to step in and assist. Okay? All right, enough of that unhappy subject. So we work gloved, but when we're wearing gloves, we don't touch anything that isn't routinely disinfected. We don't touch our faces. We don't touch our glasses. Do I have to work hard at that one? We don't touch our hair. We don't touch microscopes or textbooks or cell phones or computers or door handles. If you need to touch things, you take your gloves off and then put on a fresh pair when you go back to work. Now, sometimes you'll see people in micro labs and really in all kinds of healthcare labs doing what's called clean hand, dirty hand technique. I do this sometimes. Sometimes we're working with materials on the benchtop and we're trying to enter data into a computer at the same time. And there's no way around that. So if you're in that situation, you simply keep one hand gloved and one hand ungloved. And you just have to be careful that your gloved hand is touching all of the potentially contaminated material and your clean hand is touching the computer. One last thing to know about gloves. Gloves are considered non-contaminated waste. Now that may seem strange because we've just talked about how we're always supposed to think that our gloves have microbes on them. But the truth is, even if our gloves have microbes on them, those microbes have no way to survive very long because gloves are too dry to support the life of a microbial cell for very long. So even if we are handling culture material and things like that, and we have a really good reason to think our gloves have microbial cells on them, when we take them off, they go in the regular trash. All right, we do not routinely autoclave gloves in a microbiology laboratory. All right. Here's something you're going to hear me say a lot. The human body is covered in microbes. The surface of your body is covered in microbes. The inside of your body, any of the hollow spaces inside of you, are full of microbes. The environment that you are in is loaded with microbes. They are on us, they are in us, they are around us. Okay? When we're in a micro lab, we are purposefully creating cultures of microbes, pure laboratory cultures. We are growing microbes on purpose in containers. Okay? The other thing is, we are usually in a healthcare setting dealing with tissue samples or body fluid samples, blood samples, urine samples, all potentially full of microbes. Microbes, 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 they are all around us. How can we be successful in the micro lab? We can remember this. The two most important things to keep in your head when you're working in a laboratory that handles microbes. Number one, do not contaminate the microbe culture or the patient sample with your microbes. And number 2, don't contaminate yourself with the culture or the diagnostic sample. 2 rules. Don't contaminate the culture with your microbes. And number 2, don't contaminate yourself with the culture. Keep those things separate. If you can keep those things in your head while you're working in a microbiology lab, you'll be all set. I have to be careful because I'm covered in microbes. I have to be careful not to get my microbes into this culture that I'm growing in the laboratory. I have to be careful not to get my microbes that I'm covered in into this urine sample that was collected from a patient. I have to keep me out of those things. I also need to keep those things off of me because they are containing microbes or potentially containing microbes. Protect your materials and protect yourself. All right. All right. Does anybody have any questions? I've gone through a lot of material, I know. Unfortunately, these are the kind of things that if we were in laboratory together, we could talk about again and again as we worked in the laboratory. But because we're remote right now, we don't have that hands-on component. And I can't help you learn this sort of skill set while you're working each day. So at least right now, while we're remote, we have to learn it, on slides, which is a little bit harder. But that's why I do try to repeat things. It might drive you a little crazy, but you'll hear me repeat things quite a bit because I want to drive those things home in your memory. So Lacey's saying it feels slightly wrong to be eating a muffin right now. I know I've got my coffee, Lacey, so I know exactly what you mean. It gets a little strange, but yeah, you're only around your own microbes right now, and you and your microbes are living a happy life together, so nothing to worry about there. All right. So a couple of words about biosafety levels. This is not something that you are expected to become expert on in this course, but I do want you to be aware that when laboratory work involves living things, now remember that might be microbes, microbial cells, or that might be some other kind of cell. Some laboratories, for example, work with cell cultures, and those laboratories fall into this group as well. There are international standards that are applied to these kinds of laboratories that are there to help keep the lab personnel and the public safe. These are called biosafety levels or BSL levels, and they go from number 1 to number 4. So a BSL 1 designation means that that laboratory is able to work with the lowest level of biosafety, the kinds of things that we've been talking about today. They are not handling dangerous types of living things. From there, it goes up to BSL 2, then BSL 3, then BSL 4. BSL 4 is the highest level of biosafety. And that is a designation that is not common. But there are BSL 4 laboratories in our country and around the world. And they are doing the most dangerous, most hazardous work when it comes to working with microbes and working with other cellular materials. One of the things that I hope we can talk about a little bit this summer, even if it's more on the discussion boards, is the ongoing pandemic. Right now in the news, if you're tuned into the news at all, and I don't blame you if you're not, because there's a lot of crazy in the news, but there has been renewed interest in doing an intensive study about where the SARS-CoV-2 virus came from. we already have good indication that it emerged from a bat. We already have good indication that the genome or the genetic material inside of the coronavirus is highly matched with a bat coronavirus that we've known about for some time. So scientists believe that this virus is naturally occurring, that a bat virus underwent mutation and jumped from one species into another. That happens in nature. That happens more frequently than we would want to think in nature. So the belief is that this coronavirus that has brought the world to its knees was originally in bats and made a jump through mutation to humans. But we have not been able to prove that scientifically because the virus first emerged in a part of the world where there's a lot of governmental restrictions, and that has prevented us from doing the kind of work we need to do to really prove where this virus came from. And of course, there's a lot of conspiracy theories that say that this virus was made in a laboratory. It was man-made. I can tell you that there is no evidence that this virus was made in a laboratory. However, that does not mean that this virus wasn't brought into a laboratory on purpose to study it, and then it got out. That does happen. There are BSL-4 laboratories all around the world, and there is a BSL-4 laboratory in the part of China where the coronavirus first emerged. And if you're not familiar, it's a city called Wuhan. We don't have any evidence yet that that virus escaped from that laboratory. But scientists would like to do the work to prove that. Scientists would like to see the records from that laboratory, and they would like to know how that laboratory managed its biosecurity around the time that the coronavirus first emerged. So only time is going to tell if that work is going to be able to be done. But we know that the BSL-4 laboratory in Wuhan, China, was handling coronaviruses. So people just want to know, was this an accident? Was this a case where a naturally occurring virus accidentally got out of a laboratory? BSL-4 laboratories have the highest level of biosecurity, but accidents happen. So only time is going to tell us if that's what happened, if this virus, which occurred naturally, was taken into a laboratory in order to study it, and then it accidentally got out. Only time is going to tell us that. So you don't need to know anything other than what I've said here in terms of biosafety level. 1 is the lowest level, 4 is the highest level, and there are 2 designations in between. I do want to just let you know, though, what kind of things happen in these different levels of biosecurity. A BSL-1 laboratory, and all teaching laboratories are at BSL-1. These are laboratories that follow what are considered standard lab safety practices. So everything we've talked about today is BSL-1 level of security, biosafety. Things like, keeping your personal things away from your workspace and disinfecting your workspace and being careful when you're wearing gloves, not to touch things, and pulling your hair back, and all of those things we've talked about today are considered standard lab safety practices. And as we talked about today, the kind of PPE that we wear in a BSL-1 lab varies. It depends on what you're doing that day in that laboratory. So all teaching laboratories and all research laboratories that are handling things that are considered living things again, that are considered low hazard are at BSL-1 level. Now, what about BSL-2? What happens to make a laboratory BSL-2? In a BSL-2 lab, they're going to follow all of the same safety measures that we follow in BSL-1, but they are handling living things that are just slightly more dangerous. So they are also going to do things like limit access to the laboratory. Only certain people are allowed in the laboratory. They're going to use hoods more. Those, you've seen chemical hoods where you work inside this controlled space where the air is sucked out so that you don't get exposed to it. There are also biological hoods that are designed to allow us to work in a small space with living things. They're going to be very careful how they separate out their sharps in that laboratory. Anything that touches the living cells is going to be separated from the other sharps in that laboratory. They're going to have certain procedures to contaminate their waste. They're going to use more PPE because they're handling more hazardous organisms. These are laboratories that handle non-respiratory pathogens. Non-respiratory. Respiratory pathogens are the most dangerous pathogens because they can be transmitted most easily. The other thing to know about BSL-2 is that these are non-respiratory pathogens where we have a treatment for them. We have a vaccine perhaps for them. In other words, if there was an accident in the laboratory and somebody got exposed, they're not likely to die because there is a treatment for it or there is a vaccine for it. Okay, BSL-2 labs are not handling very dangerous things. We have a BSL-2 laboratory. In fact, we have two of them at Keene State. And one of the laboratories is using human cells. It's not a microbe laboratory, but it grows human lung cells to do toxicology experiments. And because there's always a chance that those human lung cells might be contaminated with a microbe, they're at BSL-2. Okay, so BSL-2 labs, very common and not high hazard. Now, what about BSL-3? What makes a BSL-3 lab different from a BSL-2? These labs are handling respiratory pathogens. So BSL-2 is not handling a respiratory organism. BSL-3 is. But again, there is either treatment or vaccines available. They're going to use more extensive PPE. They're going to have more extensive decontamination procedures, but really they're just following everything that happens in a BSL-2 lab, plus a little bit more. Laboratories, for example, that handle influenza are BSL-3 labs. Listen, influenza is a dangerous organism, right? It's a dangerous organism. It makes people sick. It kills people. But in a BSL-3 lab, you can handle influenza because should an accident happen and lab personnel get contaminated, there's treatment available and they've been vaccinated. They've been vaccinated against influenza. Now, the highest level is BSL-4. In a BSL-4 lab, they are handling respiratory pathogens that have no treatment and no vaccine. So all of the work that was being done with the coronavirus, at least early on, was being done in a BSL-4 level. All of the work that was happening, all of the research, all of the studies that were going on were happening in BSL-4 labs, because at that point, at least, there was no vaccine. And it's a dangerous, deadly pathogen. All right, now, I don't need you to learn this list of BSL-2 restrictions. I don't need you to learn the specific things that BSL-4 does differently than BSL-3. I just want you to know what the designations mean in terms of the level of biosafety. BSL-4 labs are the highest level of biosafety that we have. Now let's take a look at a couple pictures here. This person in this picture here is working in a BSL-1 lab. This is perhaps a teaching laboratory or maybe a research laboratory. You can see that she is wearing some sort of a scrub coat or something, or a lab coat. She's gloved. She's got her hair pulled back. She's working on a desktop. She doesn't have any PPE on other than her gloves. Okay, no respiratory protection. She is wearing glasses, but those are not necessarily eye protection. Basic work without with no particularly hazardous organisms, pathogenic organisms. This person over here is working in a BSL-2 laboratory. She's working in what we call a biological hood. This is different from a chemical hood. The idea of this hood is not to keep you safe from fumes. It's to provide a space in here where you can work with the microbe and the microbe cannot get out. So there's airflow in here that is preventing any microbes from escaping this hood. You'll notice she's got a lab coat on, she's got gloves on. Again, she has some PPE, but the primary difference is that she's now working with something pathogenic. This person is not working with a pathogen. This person is. Hey, this person in this image is doing something wrong. What has she forgotten to do? What do you think? Let me pull it up again. What has this young person forgotten to do? Here she is sitting at her hood. She's got her lab coat on. She's got her gloves on. Yeah, you've got it. She forgot to tie her hair back. That's right. Oh my gosh. If I was her supervisor, I would let her have it. She forgot to tie her hair back. Don't do that. Tie your hair back. All right, next picture. This person is working in a BSL-3 lab. This person is working in a BSL-4 lab. Both of these laboratories are going to handle pathogens. The difference is that inside this hood, this is a pathogen that perhaps we've been dealing with for a while. There are treatments available. There might be a vaccine available. She is still highly covered in PPE. She's still really doing everything to protect herself. She's working in this biological hood to keep that microbe away from herself, but she's breathing room air. If something were to happen and she were to contract an illness from this microbe, she is not likely to die from it. The big difference with BSL-4 is this. This person is in a self-contained suit that has its own supply of air. Even though she's still working in a biological hood, the danger of this microbe makes it so that she must protect herself from that microbe as much as she can. So she is in a suit that allows her to be in a completely separate environment while she's in that lab. Air is being pumped into that suit. So she is not breathing room air. And if this microbe were to get out of this hood and get into her body, it would likely harm her or kill her. So there are a lot of BSL-3 laboratories in our country. There are not that many BSL-4 laboratories in our country. I can tell you, though, that there's a couple of BSL-4 labs in Massachusetts. So they're relatively close by. All right. Questions about biosafety level. Again, something to be familiar with, so that if you were at your workplace and you were told that there was a biosafety level 4 laboratory in the building, it's an important thing to know, right? Not that you're going to go in there, because they wouldn't allow you to go in there, but it's a good thing to know. You should know what your hazards are at your workplace. All right. So regardless of BSL level, always stay focused on those two goals I mentioned to you. Keep the culture or the patient sample or whatever it is that you're working with that has microbes in it. Keep it from contaminating you. Protect yourself from that material. And number two, keep yourself from contaminating that culture or that sample material. It's got to go both ways because microbes are so, so common. Books, backpacks, purses, coats, keep them out of your workspace so you don't accidentally contaminate your things. Clean your workspace before you use it and after you use it. Use a disinfectant. Paper towels that you use, by the way, to wipe up that disinfectant, they go into the regular trash. You do not have to handle those as hazardous items. Wash your hands at the beginning of your work shift. Wash your hands before you leave. Wash, 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 wash. Questions? When I was a young veterinarian, I took a lot of chances in laboratories. I took a lot of chances that looking back now, I wish I had not. I didn't always protect myself to the level I should have. And that's because I got busy. And I noticed that some of my colleagues, they didn't do those things. So maybe I shouldn't do those things. I didn't want to appear like I was some kind of a, frightened person. Don't make that mistake. Protect yourself. Even if one of your colleagues isn't exactly being safe, do it for yourself. You're worth it. You are worth keeping safe. All right. So that really is all we need to talk about with regards to biosafety. I'm going to skip over these things because this really isn't as important to us. There's something else I want to mention today, though, because this is going to impact the techniques that we start talking about the next time we get together. When it comes to the first of those two critical things I've asked you to remember about not contaminating yourself and not contaminating your microbe culture, it's really important to do this. The truth is that in a course like this, if we were in the laboratory, we would be in a BSL-1 lab. So all of the microbes that we would be handling would be non-pathogens. They would not be able to cause disease in us. Remember that most microbes in the world, right? Remember from lecture, most microbes in the world are not pathogens. The estimate is only 1 to 5% of primarily bacteria and viruses that are pathogens. But when you're in the laboratory, you treat all of the microbes as though they were pathogens. Now, why? Why would I do that? Well, that way, if I should happen to come in contact with a pathogen, even in a BSL-1 laboratory, I'm going to be handling it safely. So we always act as though we were handling the most dangerous microbe in the world. We always handle it with that mindset so that we are as careful as we can be. Here's a couple other things to note. Microbes don't fly. Microbes don't fly. None of them. So you cannot contaminate, for example, yourself by simply opening up a culture container. You cannot contaminate yourself by simply opening up a petri dish that has bacteria growing in it. Can't do it the microbe cannot jump up off of that dish. But microbes do fall. They don't fly, but they do fall. So if I was going to open up that dish, I would want to be very careful not to keep it open very long, because microbes that are in the air, and remember, there are microbes in the air all around us all the time, they are going to be able to fall down into my container. So when I'm working in the laboratory, it's my job, anytime I'm opening a container that has microbes in it, it's my job to keep that container open for as short a period of time as possible. Because if I don't, I'm going to contaminate it. Yeah. It's going to get contaminated and my work is going to be spoiled. My work is going to be ruined. So microbes do not fly up into your face from containers. They don't do it. But they do fall down out of the air. So anytime I open a container, I need to be very aware of that. I want to keep the microbes that I'm growing in my container, in my culture, as we call it. I want to keep it pure. I want to keep only my microbes in there. So I have to be careful not to allow things that are in the air or things that are in my hair or on my skin. I need to be careful not to let those things fall into my culture containers. All right. So the other thing that we're scheduled to talk about today is microscopes. We don't have a lot of time left today. We've got about 20 minutes on the clock. I mentioned to you that I posted a document on Canvas called a microscope tutorial. That tutorial does two important things. Number one, it's a picture of a very common, very ordinary light microscope. And it's got all of the important structural components labeled for you. So all of the pieces and parts on the microscope that you ought to know. The other thing that I want you to notice in that tutorial is there's a little section about how to focus a microscope. Now that may seem like a silly thing to even talk about, but when you are learning how to focus a microscope, the best way to do it is to do this set of steps each and every time that you try to focus an object under a microscope. So there's a list of those steps, those 4 or 5 steps that I want you to learn about focusing a microscope. The other thing I want you to be able to do after looking over that tutorial is tell me, if it were on an exam, how the light comes up through that microscope to your eyes. What is it passing through to get to your eyes? Again, that may seem like a sort of a silly thing, but it's important that we know, at least on a basic level, how our microscopes work. It's important that we know what devices on a microscope allow us to do things like add a little more light, take a little light away, focus the light a little differently, change whether I'm looking at a bright background or a dark background, change how magnified my image is. You don't have to be an ocular physicist to use a microscope, but you should know the name of all of the structures on the scope. You should understand how light travels up through the scope to your eyes, and you should know the basic steps of how to focus a scope. One of the, I would say the most common thing that I hear from students when they are working with microscopes is, I can't get this in focus. And it's so frustrating for them In the micro lab, you go through a lot of work to prepare specimens to examine under the microscope, and then you get there and you can't get it in focus. And it's very frustrating when you're learning. But I promise you, if you learn the steps on how to focus the microscope, and you do those steps each and every time, you'll get good at it. Now, we can't practice this summer. We can't practice with actual microscopes. But what we can do is try to learn the steps so that when you are back in a laboratory and you are back faced with a microscope, you'll have the tools that you need to bring your samples into focus. Yes, Lacey, let me repeat those things. What I want you to know, and you can find this in that tutorial handout, I want you to know the names of the structures, the labeled pieces and parts on a microscope. And there's an image, there's a picture of a compound microscope in that handout that labels the base, the arm, the stage, the coarse adjustment, the fine adjustment, the objective lenses, the eyepieces. Those are the things I want you to know the name of. I'm going to talk about those things all semester long, and I want you to understand what I'm talking about when I mention them. The condenser, the iris diaphragm. Those are things you ought to know about a microscope. I want you to know the names of those structures. I want you to know how light comes from the bottom of the microscope, because that's where it's coming from, up through all of those pieces and parts to get to your eyes. I want you to know where the light is passing through. And the third thing I want you to know is how to focus it. And there is a list of steps in that handout for how to focus a microscope. Now, in order to make your job a little easier, you have that tutorial handout, but I'm also going to post on our YouTube channel a video. It's about 20 minutes long, showing you all those things I just said, showing you in a video the pieces and parts of a microscope, how the light travels through the microscope, how to focus the microscope. So you'll have a written version in that handout, and you'll have a visual version in the video. Okay. Now it is an extra video you need to watch, but I think you'll find it very helpful. I know for me, I'm a very visual learner. I'm a very visual learner. So it really helps me personally to see it. And I know you can't touch a microscope right now because we're remote. So that's why I've got both this tutorial for you and the video as well. Okay. So Stephanie, there is nothing on the tutorial page that says the light goes from here to here to here to here to here, but there's a picture. So knowing that at the base of the scope, in that picture again, at the base of that scope is the illumination source where the light comes out. I want you to, using that picture, tell me, okay, well, it goes up, it goes through the condenser, it goes through the iris or the diaphragm. I think it's called a diaphragm on the diagram. Then it must go up through a hole in the stage. Then it goes into an objective lens. Then it goes up through the tube, the binocular tube, and then it goes up into the eyepieces. So you're right. There's nothing on the handout that says that, but I want you to be able to do that. I want you to learn the structures and where they are so that you can trace the light beam all the way up to your eyepieces. You know, I hear people say things sometimes like, I don't need to know how a car works in order to drive the car. Or I don't need to know how my computer works in order for me to use my computer. And the truth is you don't. but your job is going to be so much easier in any work that you're asked to do with a microscope if you understand some very, very basic things about the microscope. And it's really important that you know the names of the pieces and parts. Okay. So I'm going to post that video up on YouTube as soon as we're finished with class here. I didn't post it yet, but I'm going to put it up there right after class. All right. Questions that I can answer. Any questions about biosafety? Any questions about BSL levels or anything that I talked about regarding safety in the micro lab? Anything I talked about regarding anything we've discussed today? All right. If any questions come up when you watch that video about microscopes, just jot them down. We can talk about them. Don't forget you have a quiz due tonight by midnight. You have a quiz due Friday by midnight. And you have your very first homework that you should be able to do now. And that's due by Sunday at midnight. I will see you again on Monday morning. Okay. All right, everybody. I'm going to let you go. Have a wonderful day. Enjoy the weather if it holds. And we will see you next time.